There we go. Okay, now it's working. Okay, first, I just wanted to mention a few things. I modified a bit the slides with so the mistake that yesterday was brought up, and some of the following ones as well. So I will send you everything that's updated after the workshop. As Eloy also mentioned, there will be a quiz at the end if there is time. We'll try to leave some time for you to do the quiz if you want to. But if you're not taking this class for credit, you're not obliged to do it, but you can do it if you want to. And we'll try to have a little 5-minute break after the first part of today's presentation, and then continue with the second part, and we should be done within these 2 hours, how long the workshop lasts. Okay, so yesterday we finished by getting the Trinity pipeline output, which was the Trinity FASTA file that looked like this. So we had the first line, which had information about our transcript. So it had the Trinity ID, it had its length, and then some more information about how it was actually made within the Trinity program. And in the orange box below, you can see how it looks like. So you have the Trinity name, then some identifier, C for cluster, gene, and isoform level as well. So what we're going to do today, we're going to look at how good our assembly is by using some different software to quantify that. So once your assembly is complete, you'll want to know how good it is. And you might want to compare the quality of this assembly to similar assemblies generated by alternative assemblers. As I mentioned yesterday, Trinity is not the only one that can perform the Novo transcriptome assembly. And some general ways to characterize the quality of your assembly are following. Something called the contig NX statistic, assessing the read content of the transcriptome assembly, representation of full-length reconstructed protein coding genes, trans rate, BUSCO, and detonate. So we will go through these in the next slides, but there is a link that I put below, again linking to the wiki page of Trinity RNA-seq, and here you have some other ones that you can use as well. We actually won't cover here the BUSCO and detonate, but detonate is quite similar to trans rate that we will talk about. And BUSCO also gives you an information about how good your assembly is based on the ORTH logs from known databases that you can get online. Okay, so let's start with the contig NX statistic. So based on the lengths of the assembled transcriptome contigs, we can compute the length statistic, which is called the NX statistic. And this actually means that at least X percent of the assembled transcripts of nucleotides are found in contigs that are at least of NX length. So what this means, you can see here, you have the pipeline that we're going to use. You get information about how many transcripts you have. It's separated in total Trinity genes right here. So this is the G level, and total Trinity transcripts, this is the I level, so the last letter in our Trinity ID. It also gives you information about the percentage of GC, so the GC content, and here are some stats. So as I mentioned, it tells you which proportion of transcripts, so how many transcripts are in a certain level of length. So N10, this refers to the 10%, 20%, 30%, 40%, and 50% of your contigs. And this is the length. So it tells you the 10% of your contigs have 5,264 base pairs. And what is usually used, the traditional method, is looking at the N50, which means that at least half of your assembled bases are in transcript contigs of at least this value. So at least 50% of your bases are in the 606 base pair length. The N10 through N50 values are shown as shown here. Also kind of compute together the median and average contig length. So you have a median here contig length in this example of 288 base pairs, an average contig length of 511. And this is just information of total assembled bases. This script also computes the NX values using only a single isoform. So all transcript contigs here means all of the isoforms for a particular gene. But sometimes you have a lot of isoforms, and maybe this information is not that accurate because of that. So it reports also an isoform level, which we will see when we do our hands-on practice. Another thing that the contig NX statistic can do, and actually a better way to do it, is to look at this X, it's called XN50 statistic. It's a different pipeline here, as it's shown on the top. And here, this N50 statistic is computed as above. So similar to what we talked about, the normal length statistic, NX statistic. But it's limited to the top most highly expressed genes. So it's not really looking at everything, all of the genes, but just the highly expressed genes. Because when you see here, one of the graphs that you can get out after pulling out this data is that this is your expression. So most of your genes, the highly expressed genes are here, around 90%. So we have here the percentage, the average length, and this is the number of genes inside. So what was here mentioned is that the N50 statistic was 606 length. So we have it right here. So 100% of our genes are in this category of 606. But actually, this is not a good representation because a lot of these genes are lowly expressed. So those are these right here. So when you go up to this 90%, you can see that the length, the average length of the isoform really is much, much higher. So this is like 3.5 kb for your transcript, which you could expect for a normal, let's say, gene, all the exons all together, the sequence. So it's a better way to look at how good your assembly is. Another thing that we can do is assess the read content. And this is performed by practically aligning your reads back to the transcriptome that you made. So the transcriptome assembly that you made in Trinity. These how it's done, it's shown right here. So first, you do a bow tie index that we will talk about. You input this command, and then you look at the alignment statistics. And this is the output that you get out. So it's a lot of different numbers. I want to go through them so you understand what they mean. So we have this number of reads. Of these, all of these reads, it gives you information how many are paired, if this was a paired end sequencing. And ours was, although this is another example. So we have 100% of our reads are paired, which is good. Out of these paired reads, 23% aligned concordantly 0 times. So concordantly, and discordantly are two expressions here. Concordantly refers to when two reads, so you have read 1 and read 2, which are practically the same, so it's a pair. They align in the same region of your assembly. If it's discordant, it means that they aligned in different regions for some reason. Maybe because they were not too specific, or the reads are not exactly the same matching, so they just align to a different part of your transcriptome assembly. So we have concordantly, so 0 times. This means that they didn't get aligned at all to the transcriptome assembly. These aligned only one time, and they can also align multiple times depending on how good or bad your assembly is. Ideally, they would all, of course, be aligned one time. Then you have from these that did not concordantly align to anything, the software looks whether they may be aligned discordantly, as I mentioned before. And a portion of that is discordantly aligned, but not all of them. And then when you take the portion that's left from these that are not included in the discordant, then this is the data that tells you if only one or the other pair actually aligned. So we have some of these that aligned 0 times. So this means that really these didn't align to anything. These ones aligned 1 time, and these ones aligned multiple times. And at the end, it gives you information about the overall alignment rate. So you would expect for a good assembly in this process that you would have something above 80%. So anything that's below 80% probably would need some reconsideration, maybe cleaning the reads more or changing some parameters to do your assembly. Okay, the next thing we can do is look at the protein coding genes and whether they're full length or not. And this is actually quite useful for many other reasons, not just doing it here, is because you learn how to do BLAST locally. So I assume that most of you, if you are working with genes or any type of sequences, you visited the NCBI BLAST website where you can perform different BLAST alignments. But this is actually quite useful because you can do it also on your computer directly. And in BLAST Online, practically what you do is you have a sequence and you're interested with whether any of the online available databases have your sequence or how similar that sequence, your sequence is to those that are available. But if you have a database that is yours, let's say you have your genome or you have some RNA-seq sequences that you want to use that as your database, and you want to blast something else against the database. This is not really done online because you cannot create your own databases, but here you can, and that's really, really useful. So practically how this works is that you download a database. In this case, it's UniProt website. So protein sequences, this is the biggest protein sequence database available online. You download it, you create a database using this command, make blastdb. You just define which type it is, so it can be either protein or nucleotide. And then you perform the blast also locally on your computer. So you have BLASTX, BLASTN, tBLASTN, all of the ones that are available also on the BLAST website, of course. You put in your query, so this would be your what you're trying to use. So what you're trying to align. This is your database and just some output information. So this would be the output file. You can determine the e-value, so what the threshold will be, the p-value. You can also here assign how many CPUs you would like, how many sequences you would like it to pull if there's more aligned. You can choose different ones, and also you can choose a different format of how you want this document to be presented, and I will show you this as well. And then you just run a command that will give you information about whether any of your transcripts from your assembly match the protein sequences, in this case within UniProt, and it gives you some information about where they are, which protein it is. So that's also quite useful, and we will talk about it more in our hands-on workshop. Another one is called transrate, and transrate is practically a software developed by a lab at Cambridge that is really good at kind of understanding how the assembly quality is, and it does this in a few different ways. So it inspects the contig sequences, it maps the reads to the contigs, and then it inspects the alignment. So it does number 1 and number 2, what we talked about in the beginning. And then it also analyzes the assembly by aligning the contigs against proteins or transcripts from species. So practically all 3 parts that I mentioned before, it has a different methodology, but it does the same thing. And then it gives out some metrics that we will talk about later when we actually get our hands on practice. And with this, I would like us to try this out. So we're gonna do some hands-on practice now. I would like to ask you to open your terminals. I will have the same setup as I did yesterday. So I will just put this down and I will have my terminal here. And the worksheet that I sent you guys. So let's log on first to the Hoffman using SSH, your username, and then followed by the Hoffman. If you're using PuTTY, you can just reload the session from yesterday. And then we type in our password. And we are here. So we're going to do the same thing what we did yesterday. We're going to ask for an interactive node by doing QRSH. So we're going to do QRSH -L HRT equals, we can ask, let's say, for 2 hours. And we're going to do 2 gigabytes of data. So H_data equals 2G. And then you press enter. Feel free to ask questions if you have any in the chat, or just like this. I have my chat open here, so I should see it as you're asking them if you run into any issues or if you have any questions. So this will take a few moments. And we're gonna move here. Okay, has everybody managed to get the interactive nodes? Let me just check. Okay, not everybody got their thumbs up, so I will just wait a little bit longer in case you need a bit more time. Okay. So let's start then. First, if you press LS, QRSH takes you to your home directory. So we need to get into our RNA-seq workshop folder. So we do that by change directory, RNA-seq III workshop with the tab you can auto-populate. If we press LS, we are in our workshop directory. So we have our FASTQ files that we used yesterday. We have our concatenated files that we used. We have our Trinity FASTA. This is another one that I added that I wanted to show you, but you don't have this. And if you use the link that I sent you yesterday for the drive, there are these files there as well, but you won't need them to run it here on the terminal. We have this folder that we're going to use later. And this is the Trinity out directory that was created when we started the Trinity run yesterday. But if you look into this Trinity out directory, it's not really complete. There should be much more files if the run was finished. If we didn't stop it, you would have a lot of more files here, including this Trinity FASTA file. But let's go out of there, back to our workshop folder. Okay, so we have our Trinity FASTA. We can have another look at it to see how it looks like. So if you do head Trinity FASTA, we have all of our contigs, our transcripts right here with their names, the length, and the path. So first thing we want to do is to look at that contig NX statistic. So because we worked with the variable Trinity home yesterday, the software didn't actually remember it. So we will need to do this again to create this variable so we can use it for the rest of today's workshop. Again, we need to get the path to the Trinity home, and I don't remember it by heart. So let's look where it is. We know that it's in software. So it's right here, Trinity RNA-seq. We need to go into that. So we go CD tray tab. Okay, so it's right here, this Trinity. And we're going to be going into these folders as well, but this is going to be our variable. So we're going to create the variable here. So pwd, print working directory to get the path. We copy it, and then we write export Trinity home equals, and we paste this. Enter. So now if everything worked correctly, we can just quickly check if we write Trinity $sign followed by Trinity home. You should get this bash path is a directory. So we created it the right way. Okay, so now let's go back to our workshop folder. So we move up, CD. This is 2 dots. This is one directory up. So this would be software. We need to go one more. So you can just do this. Okay, let's go 2 directories up. Enter. LS. And we're here. It's written right here. RNA-seq III workshop. Cool. So now we're going to run the Trinity stats pipeline. So we go Trinity home. And this is going to be within Trinity home, another folder called util that we looked at yesterday briefly. And inside there is this pipeline, Trinity stats.pl. Then space, and we just say which assembly we wanted to run it on, which for us is this Trinity FASTA. And then you press enter. And it's pretty fast. Okay, so what do we have here? It tells us total number of Trinity genes. We have 2,386. So this is really, as I mentioned yesterday, this is just a subset of the data that we're using. So some of the things were going to be much smaller and they might not even work because it's not a real data set. Then we have total Trinity transcripts. Again, this is the gene level. This is the isoform level. So we have some genes that have multiple isoforms because this number is bigger than this one. And the GC content is 53%, 53.37%. Then what we talked about before, we have stats based on all transcript contexts. So this is including all the isoforms for each gene and the NX statistic. So at least 50% of our basis is within contexts that are at least 416 long, which is not bad. Of course, you want this number to be as big as you can. Then we have the median contact length. That's a little bit smaller. We have average contact length is 400, and these are all the assembled bases that it used. But then when we go down, there's another portion right here that talks about only the longest isoform per gene. So you can have isoforms that are different lengths, of course, and this is just taking into account the longest one. Again, the same statistics, and here you can see that the N50 is a little bit actually smaller than this one here. So you have slightly, in this way, you have slightly less bases so the average contact length for at least 50% of the bases is 393, and we had 416 before. And again, it reports the median contact length and average contact. Okay, so this gives us a little bit of information. It's very informative, but not really. We cannot conclude much about our assembly. Just we know a little bit how many genes we have, what the basic statistics looks like. What we're going to do next is look at the read content. If you remember from my presentation, there was another step here with the contact NX statistics called XN50. To be able to perform that, we need to do the abundance estimation first, because it uses the abundance estimation to be able to detect those highly expressed genes. So we cannot run it just yet. So we're going to move to the next part, which is assessing read content. First thing we need to do is build an index for the Trinity FASTA. We're going to do this by using bowtie 2. So index is practically an index, a list of features that helps bowtie build the alignment much faster. It's kind of similar to what you have in a book. So there's an index, so you can actually know where things are within a book. It works the same way. We need to load the bowtie module first, which is inside Hoffman already. So that's easy. We just do module load bowtie 2, enter, and now it's loaded in to our path. And the next thing we do is build the index. So this is the command, bowtie 2 -build. Trinity FASTA. So the first one is our assembly, and the second one is the index. We're using the same name. It's just building it under the Trinity FASTA gene file, but it's not going to change our initial assembly file. And then we press enter, and this should run pretty fast. When you have a lot of genes, a lot of reads, this takes some time, so it does build it quite slow. But because we have just a portion, like 10,000 reads per sample, this runs pretty fast. Okay, so if we press ls, you're gonna see some more files here. So this is our original assembly. And then we have this Trinity FASTA BT documents, and this is actually the index itself. It's within these documents, these files, and Bowtie. In the next step, we'll use these files to be able to easily find whatever it needs to find within the assembly. Okay, so now we're ready to align the reads. Oh, cannot open Trinity FASTA. Just try to make sure that you're putting that you're in the right directory, and you can copy paste this to make sure that you are writing it correctly. So you need to load in your Bowtie first and then do this. Maybe try to just make sure that you're in the right directory. If it's not working, just send another message, then we can troubleshoot through it. We will slowly move on so you can catch up in case you're... okay. So just make sure you're inside the RNA-seq III workshop, and then it should work fine. Okay. So the next thing we need to do, as I mentioned, is the align these reads. And we use just Bowtie2 as the command, followed by some parameters. So -p refers, this can be changed. This is just how many threads you want this to run on. -q refers to the file type. So whether it's a FASTA or a FASTQ file, we have FASTQ files. No on al is just telling it to when it creates a SAM file, not to include the ones that are not aligning properly. -k gives it information about the minimum length that it wants that it can use, and this is what we have to input. So -x is our assembly, which is Trinity FASTA. -1 is our left read, so we are going to do all our one FASTQ, and -2 is all R2 FASTQ. This will redirect this pipeline into comprehensive align stats text file that we will look at, which will give us information about how good our alignment is, followed by another tool called SAM tools, and this is just going to create a BAM file, so an alignment type file from this, because this is going to create something called a SAM file, and a BAM file is the same as SAM file, but it's a binary file, so it's not really readable by us, but this is something that you can create to use later for any type of alignments that you might do. So let's type this in. We can copy paste it, but let's practice. Bowtie2 -p. Oh, before we start, we need to do something else. We could have waited to get an error, but we can also do it beforehand. So SAM tools is another tool that we need to load in, and it's not going to work without it. So actually, let's do it. Let's do it without, and then we can just load it in. Bowtie2 -p 10 -q for FASTQ. No. UNL -k 20 -x Trinity FASTA 1. We're going to have our all read 1 -2. We're going to have our all read 2. Then we're going to redirect this to a new file called align stats.txt. pipe. Pipe is a very useful tool because if you've taken the Intro to Unix or you are familiar with this, it's a nice way to combine different commands together. So the one that was before is the input for the other one, and you can put as many as you want. And then we have SAM tools. View is just to create a SAM from BAM or BAM from SAM. Again, some threads. And we're going to have an output of called bow tie to BAM. Let's hope I didn't make any mistakes. Okay. So it's not running because SAM tools, as I mentioned, is not loaded in. So we need to load it in before running. And this again is incorporated in the Hoffman cluster. So we just do module load SAM tools like that. And then if you go arrow up, find it again. And if I didn't make any mistakes, this will run it and create an align stats file that we will look at. This might take, oh, it's already done. So if we do ls, it should be right here. To look at this, we just use the cat. So cat can be used for concatenating, but it can also be used to look at your files within the terminal. So if we look at our align stats, there we go. So we have something similar to what we observed in the presentation before, although numbers are different. So much less reads, you know, a certain portion aligned concordantly, discordantly, some aligned as singletons or some didn't align at all. And this is what's important here. So 47% overall alignment rate. This is pretty bad. So this is, if you get something like this, you would probably think about, okay, I should change something in my reads. Maybe there's a lot of low expressed reads that are not aligning well to my assembly, and my assembly was 2 is just like it's a way to tell it to create this file. It should work also without it as well. This is the pipeline that they use. I think this should work without 2 as well. So it's really, this is really bad. So you would, if this happened to your full alignment, full assembly, you would definitely reconsider. Because we're using only a subset of data, and this data is not really controlled for high or low expression. It's not really surprising that the alignment rate is bad, because we're only using a super small portion of the data that probably a lot of them are lowly expressed genes. Cool. So now we have our alignment rates. What else can we do to check our assembly quality? We can do the local blast, as I mentioned. So to do this, we can download the Blast Plus, but actually this takes quite some time. So we're not going to go through this in detail. I will just show you how this data looks like. But I want us to download Blast so you can see what kind of features it has and what you can use it for in your work in general as well. So to do that, let's go into the software directory. So CD software, we have all of our software packages here. Again, you go to the NCBI website. There is a download Blast Plus link, and there should be a latest version. This is actually not the latest version, because cluster the Hoffman for some reason does not support the latest Blast. The only one that works is this 281. So I copied it, and we're gonna just use wget to get it again, because it was in 2 rows. There's a space here that shouldn't be here. Okay. So this is really useful, but I was troubleshooting this a lot because I couldn't get the BLAST to work. I found out that actually the cluster, at least for now, cannot run the new BLAST. If you ever want to do some BLAST on Hoffman, make sure you're using an older version that is going to work. That's just a good tip that I had to explore for a long time until I figured out what was going on. Okay, ls, the tar file should be here, NCBI BLAST. We need to unzip it using the tar command. So we have tar minus xvzf, extract, verbose, unzip, and F is to give it the following file. And we just press enter after writing the package name. You do not have to put V if you don't want this verbose to appear. But I like to have it just to see what packages are installed, and in case there is some error, it's going to give me information immediately here. Okay, so now we have it right here. If we go inside that directory, so 281 plus, enter. Did you copy paste this website, Alex? Yeah. Sorry, it's Sheridan. Alex is helping out. Yeah, so there's going to be a space right here between this NCBI here and BLAST because it's in the second line. So just make sure you don't have any spaces. We're gonna let you work on that in case you run into, if it's not working, let me know. Then I can show you how to get it from the website in case there's something going on, something else going on. Okay, so now we are in our NCBI BLAST directory, ls. Well, we still don't have any greens. We need some greens to be able to run where executable files are. And it's actually right here in this bin. So we need to go one level down. We go CD bin, and they're right here. So you have blast N, blast P, blast X, P blast N, all the things that you are already familiar with by using Blast Online. Okay, so the newest version is fine. You can download it here just for practice, but it cluster has some issues running it. That's why I'm downloading the older one. But yeah, of course, you should always use the latest if possible because it's going to be more up to date. Okay, so we have all of our executables right here. The one that is important to create the local BLAST database is this make BLAST DB. One useful thing that you can use here if you're not sure, okay, but how do I make this? What kind of parameters do I need? If you just type whichever of these, let's say make BLAST DB minus H minus H. Oh, okay, then I guess I need to put help. Sorry. Okay, this is also a useful tool. So it says make BLAST DB command not found. To run make BLAST DB or to run anything within a directory that you're in, you actually need to put something like this dot and forward slash, and now hopefully it should work. Okay, so it opens kind of a manual for us where we have all the information that we might need. So it tells you, okay, make BLAST DB minus H or minus help. You can put an input file, you can put an info file type, a lot of different things that you can use. The same for BLAST-P. So we all know that for BLAST-P, we need a peptide, so we need a protein sequence. But there's other things that you can specify here, as you can do on the website as well. So BLAST-P help, everything in detail written here. But if you go up to the top, again, it's giving you a lot of options that you can use with BLAST-P when you're running it here. Some things are necessary, such as of course, the database, and what kind of database is. There's a lot of things here. I don't know where it is. You can put output formats, as we're gonna go through. And then if you want more detail to actually know what all these things mean, is just here below. And regarding the output format, that's right here. So there's different ways how you can get it out. For instance, I like to use 0. This is practically all the possible combinations of alignments, and it gives you information kind of similar to what you get on the website with nucleotides, and which ones align, which ones don't, some information about the e-value and all kinds of things. So it's very, very useful in general. Okay. So what you would need to run the representation of full-length reconstructed protein coding genes within the assembly, we need some sort of database that we're going to create. So we have our Trinity FASTA file that's going to serve as the query, but we don't have the database. And the database would be a protein some sort of protein sequences that are already available online. And that's the UniProt from the UniProt website. To do this step, it takes quite a long time because it's a big database. That's why I don't think we should do it here, because it's really going to take at least 5-10 minutes, and then running it is going to take an hour. So if you would like, feel free to download this afterwards. If you run into any issues, just let me know. But the website should be right here. You can get it again with wget. It's going to be gzipped. So you would just need to unzip it by doing gunzip, like this. Gunzip, gunzip. And then after that, it's written here. And then after that, we would create again the path to be able to not have to like repeat the whole path. We create a blastable database. So after we have the UniProt, we use that to make BLASTDB that we looked at, the input, which is our file, and then the DB type, which is protein. It can be nucleotide as well if it's a nucleotide database. And what it creates is a lot of 3 files that look like this, followed by another extension that then practically serves as the BLAST database. After that, we perform the BLAST-X because we have a nucleotide sequence. Trinity FASTA is a nucleotide sequence, and we are blasting it against the protein sequence. Again, so I mentioned Trinity FASTA. We have the protein, the protein database. Even though there are extensions here in the files, when you put -db, you just put without the extensions, the name without the extensions, and you specify the output file name, the e-value, and the format. So actually, I have it right here, and I can show you how it looks like, the output. This BLAST-X and BLAST-P in general take a very, very long time. So again, you would run it remotely using QSub. But what it gives you is this. So practically for each of your Trinity gene, Trinity transcript, it tells you whether hits are found or not within the protein database. So for instance, for this one, no hits were found. For this one, no hits are found. For this one, there are several hits found. So you have the UniProt name, you have the score. So 0 would be the highest, the better here, because it means that more nucleotides are aligned. And this is the e-value. 0, of course, would be the best. And then this is out form at 0, which is my favorite because it really gives you all the information with this. So kind of a comprehensive list. And then it separates each of these, saying, okay, this particular hit in the UniProt, it has this length, the protein itself, a lot of different informations about p-values, identity, so how many proteins, what proportion of the protein was aligned, if there are any gaps, and what is the frame, so the open reading frame. And then it just aligns them one next to each other. You have the query. This is your Trinity FASTA. This is actually the translated protein from the Trinity FASTA. And this is the actual UniProt protein. And here it tells you which ones are similar, which ones are different. It's quite useful when you need to run some local blasts to do this as well for your sequences. Of course, here we have a lot, so we're not going to go through all of that in detail. But it's a really, really useful tool, in my opinion. Okay, let me just reopen this. So you would have a result from this, which will be a table with a lot of information that I just showed you. So I would kind of compile all these this information from the BLAST-X output into a table, and a portion of the table would look like this. So you would get information about the length, but also something something like this. So you would have the number of genes that are in a bin. So these are the bins from 0 to 100. This is the percentage, bin percentage. So what this tells you is that, for instance, here, there's 268 proteins that match a Trinity transcript by 80% and less than 90% of their protein length. And then also, you know, there are 3,515, 510 proteins that are represented by nearly full-length transcripts having 80% alignment. So this is practically the alignment coverage. And then something else, like there are 300 and 242 genes, which is right here, that are covered by more than 90% of their protein lengths, so from 90 to 100. So it can be quite useful just to get an idea how well your assembly is, how good, how many proteins you get, whether your transcripts actually covered the full length of known proteins, because that's what you want. You want to have a full transcript that can then be translated into a full sequence of a protein. Let me just see the question. Does the output specify which version of the protein sequences are being used? What do you mean about version? I'm not sure what you refer to by version of the protein sequence. Possibly. I'm actually not 100% sure. So it gives you this information. It doesn't specify the version, but what you can do, I guess, is to go look look in UniProt because this is a UniProt database. You can look at the ID number, if that helps, if that answers your question. Yeah. So this is all the information, and this cannot be changed because this is encoded within the UniProt file that we downloaded. But of course, you can take all of these and go have a look at it online for more information. Okay. So we have one more thing left. Let's open up this. We have the trans rate. So trans rate, as I mentioned, is a package that was developed by a group at the University of Cambridge, and it's a pretty nice tool to explore your assembly as well. So this is the website where it's where we can download it from. So let's do that. Before we download it, let's go into our software, because currently we're in this bin of the BLAST. So we need to go 2 levels up, if I'm not wrong. Software, okay. And then we do wget space and just make sure you have everything without any spaces. Okay. Where is it? It's right here, trans rate. Again, we need to unzip it. X. Let's do XZF so you see how it looks like without the verbose. So it's just gonna unzip it without giving us any information how it does it, but it should still do it. There we go. So first thing we're gonna do, let's create a variable for this as well so we don't have to input the whole path. So we go CD, let's go inside the folder, trans rate, and here it is, our executable file. So print working directory to get the path. We're gonna copy it. Export, we can name it whatever we want. You can write TR just to keep it short if you want. We put in the path. What's going on? Maybe we do need to put something longer. Oops. I don't know why this is not working. Let's try again. Export. I was putting the $sign. Okay, now I figure out what I was doing. This $sign shouldn't be here. Now it should work fine. So if we just do $sign trans rate, we should have our path is the directory. There you go. So it's really, these like easy mistakes happen a lot. So it's important to try to like look at where you made a mistake, because in most cases it's something very like mild as this one. Okay, so we are in the trans rate folder right now. Let's go back to our RNA-seq III workshop. So that's going to be somewhere up, a few directories up. So 2 ls right here. And let's run the trans rate command. So we did $sign trans rate. This is going to be our variable. We need to give it the executable file, and we just need 3 things. So we need our assembly. We put our assembly, which is Trinity FASTA. We give it left for our left files, and we give it a right for our right files. And then we press enter. And this is also going to be pretty fast. So it's calculating some metrics. It's probably going to give us an error now. Okay, cool. Let's start from the top. So it tells us what it did. So it loaded it and analyzed it, and it gave us some information right here. So it reports a few different things. First, the number of contigs that are in the assembly, the size of the smallest contig, largest is the size of the largest contig. It tells us the number of bases that it included in the assembly. It tells us the mean length of the contigs. So this is also useful because you can compare different methods, whether it's giving you similar results of doing the quality checks. Then this n under 200, it tells us the number of contigs shorter than 200 bases, and over 1K, the number of contigs greater than 1K, over 10K, greater than 10K, and with open reading frame, so the number of contigs that had an open reading frame, mean open reading frame percentage if not the whole open reading frame was covered, and 90, 70, 50, 30, 10. So this is practically the same as in the NX contig statistics. So the largest contig size at which at least a percentage of bases are contained within that contig. GC level, the number of bases that are n, so the bases that are not assigned to ATGC, and then that's it here. Then what it tries to do is to build a snap-in, so to align the file similarly to what Bowtie did before, because our data is too small. This didn't run properly, but that was expected. So if this snap would run normally, you would get an information about the assembly. So when you're doing a full assembly, it gives you something called the assembly score, and it can go from 0 to 1, so 0 to 100%. And the better that number is, the closer to 1 is, the better your assembly is. If you have, for instance, 90% alignment rate with Bowtie, you would expect to have at least like 0.8 or 0.9 here as well as your assembly score. So there are a lot of different ways to test your assembly. You don't have to do all of them. You can do more than these, but it's up to you, and it's just a useful tool to see whether you should continue with this assembly or not for the other steps that we're going to perform. So the post-assembly analysis. With this, we can take a few minute break. If you would like, you can ask questions, or if you're running behind, you can catch up. And probably like 3 minutes, so it's 10, 11, 10, we can just restart. And then we're going to go on to the last portion of our workshop. And in the meantime, I will send you all the quiz so you have it as well. And I'm going to do that right now. I will send it to you in the chat, but also on the email, so you have it for later. When I run tar for the transfer rate, I get an error not recovered. This can be maybe due to if the file was corrupted when you were getting it from the website, maybe. This can be a possibility. So if you're not sure if it persists, then try to just do RM for remove, followed by the transrate tar file to remove it. It's going to ask you, are you sure? Yes. And then try to get it again from the website. Okay, maybe we can continue. And we can, I'll send you the link to the quiz to the email later, but you have it in the chat if you need it for later. Okay. So let's continue with the presentation. There's a few more things on the slides that I wanted to show you. Okay, so once you check your assembly, you are happy with it, you think it's good, we need to process the data by doing post-assembly analysis. So what we're going to do today, we're going to do abundance quantification. We're going to do some quality checks of the sample and biological replicates. We're going to do differential expression gene analysis. We're going to detect coding regions and also talk a bit about the functional annotation. So if you have multiple sets, multiple RNA-seq data sets that you want to compare, you have to be sure just when you run the Trinity to assemble it on all of your samples, as I mentioned yesterday. And then when you run the abundance quantification, then you use your samples, your conditions separately for each abundance quantification. So any of the abundance estimation methods will provide transcript level estimates of the count of RNA-seq fragments that were derived from for each transcript. In addition to a normalized measure of transcript expression that takes into account the transcript length, the number of reads mapped to the transcript, and the total number of reads that mapped to any transcript. So what does that mean? Firstly, you can do all of these things by using different software that's already provided within Trinity. So you have R, SEM, Callisto, and Salmon. If you have done some RNA-seq analysis or abundance quantification, these might be familiar, especially RSEM. And these are actually different. So RSEM is an alignment-based method. Practically what it does is shown right here. So we have our Trinity FASTA file, that's our transcriptome as reference. And it aligns our reads to this transcriptome that we have. In a normal RNA-seq pipeline, you would have something like this. So you would have your reads and they would align to the genome as your reference instead of the transcriptome as we have here. But then we have something else called Callisto and Salmon. These are two different softwares, but they work in the same way. And these are alignment-free methods. So they don't need this genome. They don't need this transcriptome. They're just doing kind of like a pseudo alignment using k-mer abundances to be able to perform the abundance quantification. And because of this, they're actually a bit faster as well. Trinity does not come prepackaged with these, so we need to make sure that we install all of these before we run them. Today we're just gonna use Salmon, but for all of them, you would behave the same way. So the abundance quantification works on, it can work on 2 levels. So it can be run on gene and on isoform level. If you remember our Trinity FASTA file, we have our gene and our isoform level, so we can get information about the abundance for both. So this is the command that we use. We have the Trinity home, that's our variable. Then we have the util folder followed by the script. There are a few things that we need to specify. First is the transcript. So this is our FASTA file. The sequence type, again, that can be FASTQ or FASTA, left and right if it's paired end. We have this samples file that we need. So this samples file is kind of like a little metadata that tells the pipeline which sample goes for which condition. So you would have, for instance, in our samples, we have 0 hours and 6 hours. 0 hours would be condition A, 6 hours would be condition B, and then here we would have 0 hours 1, 0 hours 2, 0 hours 3. We're gonna do that later. The estimation method. So here it can be Salmon, RSEM, Callisto, whichever you're using, and output directory. So where we want our results to go to. Alignment method. This is used if you're going to use RSEM. So because Salmon and Callisto are alignment free, we don't need this. If you're going to use it, then you just specify that it's going to be bow tie 2. And the Trinity mode, this is just another part that you just put to specify that your input reference here is from Trinity. There's other, again, parameters that you can use when you run the abundance estimation, and you can find it right here on this link as well. Okay, so what is the output of Salmon, for instance? We have 2 files that we get out. We have quant.sf and quant.sf genes. So quant.sf is the transcript abundance estimates, and the genes is the gene level abundance estimates. And this is the table, one of the tables that comes out. So we have the name of the gene, its length, its effective length. So this is just a little bit lower because it does some calculation what actual length should be based on the reads that align to it. The TPM, so these are the normalized counts. Transcripts per million transcripts, that's what TPM stands for. And this is the number of reads that align to each of this Trinity transcript. Then what we need to do is so practically you do this for each of your samples. So we're gonna do this for our 0 hour and our 6 hour samples. And then we wanna just put them all together to construct a matrix of these counts and a matrix of normalized expression values. And then we're going to have everything all together in a table that then we can use later to do differential expression analysis and all the QC, so the quality checks. Once you've performed this transcript quantification for each of your biological replicates and made the matrix of this data, you're going to have these outputs. It's good, as I mentioned, to examine whether your data are actually good, whether the biological replicates represent what you want them to represent, and just to investigate the relationship between different samples that you might have. And this can be done in a few different ways. So Trinity has some scripts that can be used for this. For this particular one, you need to have the counts matrix, which you get from doing the abundance estimation, and you need this samples txt file. Something that you get out is, so this is the command that we're going to talk about later when we run it. Whoops, where are we? You get out the sum of mapped reads. So it just gives you, these are your bioreplicates. It tells you how many reads you get. It makes these pairwise comparisons of replicates. So everything that's read is different from each other on a log2 scale. So log2 difference. You have the same information, but these Manhattan plots, MA plots. And then what it also tells you is how your samples are correlated by doing this Pearson correlation. Another thing that you can do is not to look at your biological replicates. So this is done for the biological replicates within one condition. You can look at how your different biological replicates in different conditions look like. And it's practically the same strategy, but here we tell it to calculate everything. So the biological replicates and all of the conditions, and you get a heat map like this that gives you a little bit more information about it. So these here would be your conditions, and these would be your replicates right here. And the red represents more similarity, the green represents less similarity. So from this graph, you would kind of infer that these two samples, these two conditions are probably closer together, more similar than, for instance, this third one. So it's quite useful if you would expect something like this. For instance, if you have, this would be a condition, a normal condition. This would be a treatment, and this would be the rescue treatment. So that you would expect this to be similar to your condition than this one, for instance. It's a good way to check whether, it's valid, whatever you're doing. Another useful way to check the quality of your samples is to perform the principal components analysis. And here there is a command that does that. Practically, this principal prints comp 3 right here tells the software to give out the first 3 principal components. So you can have many principal components within when you run a PCA, but probably the ones that are top, so 1, 2, 3, 4, 5 sometimes represent the majority of the data. So a principal component plot looks like this. You would have your different conditions, so red, green, and blue. And when they cluster together like this, it means that the end close together, it means that the bioreplicates are most likely correct. So they're good, they're very similar, and they're apart from your other conditions, which you also would want, because you don't want the conditions to be mixed if you're going to look at some differential expression or something similar. Okay, so after we confirm that our data looks good, we can then perform the differential expression analysis. And the differentially expressed transcripts of genes or genes are identified by running this script below, which will perform practically a pairwise comparison amongst each of your sample type. And we will actually see that when the command runs, how it does it one by one. So what we need to indicate is our matrix, so our counts, the method that we're going to use. Trinity, again, supports a different differential expression software. Today, we're going to use EdgeR. And again, you can input this samples file, which tells the software which samples belong to which condition. Then the results that you get out is something like this. You have some tables with your Trinity gene. This was wrong actually in the presentation, so I'm gonna send you this new version. The log fold change, so how big the difference is, the CPMs, so these are the counts, and then the p-value, and there should be another column here that tells you about the false discovery rate, the FDR. And it gives out some nice PDF files as visual representations of your data. So a Manhattan and volcano plot, so everything that's in red would be differentially expressed in these two. So it's just a nice, visual representation of your data to help you understand what's going on. And then what else you can do? You can cluster your genes. So by running this script right here, you give it the matrix and you give it some cutoff values. So here would be the p-value, and this would be the fold change. And the output looks like this. First, you can get a correlation matrix again, but then also a heat map. So this is what you normally see in papers when they're doing RNA-seq experiments. You have heat maps like this that are separated by your samples, and it indicates any clusters, and it indicates when you have an overexpression and underexpression of specific genes or gene families. And with this, another useful thing that you can get out of Trinity is to cluster them by partitioning the genes. So each of these expression patterns can be visualized like this. So they would, a portion of genes would fall into a category, especially if it's something like a time course. You can follow a portion of the genes and their expression in time. So you would have these clusters, and then you could use them to infer some more information about, whether you have a cluster where you have only some expressed in a rescue or in your treatment condition. So it's just a really nice way to visualize these pathways as well. And this is developed in Trinity, but also can be used for your other data. If you have a accounts file, you can perform all of these as well for some other experimental designs that you have. And the last thing that we will cover in our practical is the coding region identification. So just how do we identify ORFs and proteins from the Trinity FASTA assembly file that we have, that then we can use for doing functional annotation. For Trinity, they developed a software called Transdecoder that we will use to do this. And they also developed another tool called TriNotate, which helps identify which Go terms, which PFAM domains can be found within our assembly. TriNotate is quite useful, but it's quite lengthy. So that's why I mentioned here just several steps that are required to do the TriNotate. And on this website, you can actually find a lot of information about how to run it. But practically, it downloads the databases online. So Swissprot, UniProt, as we mentioned before, PFAM for the domains. It puts them all together in this SQLite database, and then it outputs an annotation report, which practically gives you information about your Trinity gene, whether it was found in Swissprot, whether it was found in PFAM, a lot of different information. And when we come to this, I will just show you how a table would look like, a table that comes out of trinotate would look like. Okay, so let's do some practice with these things, with the post-assembly analysis now. So we go back to our screens, we check where we are. So we are in the RNA-seq III workshop. The first thing that we're going to do is the abundance estimation, and we're going to use salmon. So first thing we need to do is to create that samples file that I mentioned. And it looks like this. So we're going to use Vim as the editor, and it's very easy. So you just do Vim, and then followed by whatever the name of the file will be, which for us will be samplesfile.txt. Enter. This bash script open, we press I, and this will change into insert. So we can now start writing inside. Okay, so these are tabs separated. So we will do 0 hours, tab, 0 hour 1. Oops. 0 hours, we have 3 replicates, so we need to do this 3 times. 2, 0 hours, 0 hours 3. And then we have our 6 hour, 6 hour first bioreplicate, 6 hour tab, 6 hour second bioreplicate, and our last one. So we will have 6 rows. After we are done, first press escape. This insert will move. Then press colon. It's going to appear here below. WQ, which means write and quit, and enter. It's going to take you back to this terminal right here. LS should be right here somewhere. There we go. And if we want to look at it, you just do cat samples file, and it's right here. So 3 of 0s, 3 of 6 hours. Okay, so we have that done. Now we're going to run the abundance estimation for each sample. As I mentioned, this has to be done for each sample, so for each bioreplicate, and then we're going to pull them together later. So this is the command. Maybe we're going to copy paste these because they're pretty long. So we have our Trinity home, followed by this util. So that's a folder where we have our script. We need to input our transcripts. We need to input the sequence type, which is FASTQ, our left reads, our right reads for this bioreplicate. So we're going to do one by one. Estimation method, the output directory. So we're going to create a directory that's going to be called 0 hour 1. And this is something that you need to put in for the software to know that that it needs to run it with this Trinity FASTA to prepare the kind of like a pseudo reference for it as it's running. And then you press enter. Okay, we get an error. Why do we get an error? There's no salmon that we put into our path, so we need to do that first because we are trying to run salmon. Okay, so let's double check where salmon is. So we do cd software, and we know that it's in software, and then it should be in this salmon latest, but we need to go into the bin if you remember. So it's right here, pwd. We're going to take this. We're just going to put it in the path like we did yesterday. So we put path equals $path, colon, and we paste this like that. And then we go back to our workshop folder. So that should be 3 levels up. Yes. ls. Okay, we're back. So now if we go up with the arrows to find our command so we don't have to type it, there it is. And we press enter. And now it's working. Okay, it finished really fast. We have some issues with a potential strand bias. So it tells us, okay, maybe you should have ran it in stranded mode, as I talked about briefly in the introduction. But this is not an error. This is just a suggestion. So for now, we're going to just leave it like that. If we do ls, we see this folder right here. And if we go inside that folder, 0 hour 1, we will see that we have the quant SF. The reason why we don't have the quant SF genes is because this is really a small data set and there's not a lot of genes that it can create some sort of meaningful matrix. But if you have a lot of them, you would have the genes as well here. But we're gonna use the isoform level for later. Okay, so we have that. Let's go out back to our workshop folder. And now we're gonna do this for each of our bioreplicates. Again, let's copy paste because there is a lot to write. So we run it for the second one. Then we run it for the third one. So we finished the 0 hours. Let's do the 6 hours. So you can see here that I'm just changing the FASTQ files and the output directory name. So we have each output in a different folder. That one, and this is going to be the last one. Like so. Okay, so if we do ls, they should all be here. So we have 0 hours, 1, 2, 3, 6 hours, 1, 2, 3. And all of these inside should have the same thing. Okay, so now we quantified the abundance. Now we need to make a matrix of all of this. And this can be done in R. R can also be accessed directly through the cluster, which is extremely useful. So you don't need to have your R software separately on your computer. First, what we need to do, we need to load the module R. It's already there in the cluster. So we can just use this version right here, 361. So we do module load R 361, enter. It's going to unload and load some models that are conflicting. That's fine. To access R within the cluster, you just press R, capital R. And you see here, similarly to what you see in RStudio, if you have ever opened it, it just has this greater than sign, and this is where you write your thing, your commands. Okay, so we need to install EdgeR. To do this in R higher than 3.5, we use this BioC manager, and we need to just give it this command to be able to install it. So we can just copy and paste this in, and it's just gonna run it, and that is fine. And then after that, we're gonna install EdgeR by using this command. And now if everything is fine, it should run and install EdgeR for us. This does take a few minutes because EdgeR is a big package. So for EdgeR and for DE-seq DE-seq II, those are kind of like the two most popular gene expression software tools that are used. You can use both of them. You can use one and then the other. I think in terms of differential expression in general, it's useful to run a few different methods to be able to compare them as well, because although they are meant to perform same, like they should give out same results, they're not really always the same. They're similar but not same. Some are more stringent, some are more like broader. So it's really good, in my personal opinion, to run a few different ones, and then you can conclude which one works best for your type of data and your kind of biological question, because you should have a biological question preceding running an RNA-seq experiment like this. Okay, so while this is running, if you have any questions or comments, feel free to write in the chat. This should be done pretty fast, maybe another minute or so. And R like this is very handy because you can do a lot of things. The only thing that maybe is limiting is that you cannot look at all the graphs that you get in R, but then you can just look at them on your local computer, and we will actually do this in a second after we finish running the differential expression. What is the time? 8:41. Okay, perfect. Okay, now it's done. When it shows this, it means that it finished. Oh, we need to update all these. So it's really useful because you can access R and you can get out of R. And if you put a command on the normal terminal, so not inside R, you can kind of feed into it and just do its thing within R, and then come out and output the result for you right there as well. So sometimes it's asking for these extensions. So I pressed A for all that I want all installed, and this just adds a bit more time to how long it takes to install it properly. But it's done now for me. So when you have this bigger than sign, it means that it's done. I hope everybody has their EdgeR installed, or we can wait for a few more minutes. Okay. So if you get that message, do you want to update all, none, or some? You can just press A for all. Sometimes it asks that, sometimes it doesn't. Okay. So now to get out of R within the cluster, we need to just write quit. Quit followed by closed brackets. Save workspace image, yes. So it's gonna save all the EdgeR information that we just loaded in. Yes, enter. And it just puts us back into on the cluster. Okay, now we're ready to create that matrix. It's inside Trinity Home again, inside this util. What we're gonna put in is the estimation method, which is salmon that we used. Out prefix, this just we're just specifying what kind of prefix we want for all of the files that are going to be created, which is Trinity Salmon. It can be anything. Name sample by base directory. This just means that it's going to keep these names. The gene trans map, you can specify, or you don't have to specify if you're not going to run it on a gene level, which we're not running. We're doing isoform, so it's none. And then followed by all these quant.sf files that are within our folders. So this just means go inside this folder and get me this document for each of them, because we're going to run it from the RNA-seq III workshop folder. Again, let's just copy paste it. So we have everything here, and we press enter. And we get an error. There is none. Okay. There's a space here. You can see the command line can be very sensitive. It is sensitive to every mistake that you make. I hope that's the only one. Yes. Okay, so what did it tell us? It took all these files that we gave it. It outputted a matrix, and it wrote it in a table. It wrote it in a few different tables, actually. So if we do ls, we're going to see some new files here. We have isoform counts matrix. These are the normalized ones. The TMM is the normalized one, and we have some other information right here. So the Trinity Salmon isoform counts matrix, if we just look at the top using head, it looks like this is switched a little bit to the left, but we have the name, we have all of our conditions with the counts right here. The TMM would look a little bit different because it's normalized, so it would have different numbers, but the structure is the same. Okay, so now what we can do, we can check our biological replicates and our samples. So there is this script Peter PTR inside the Trinity that we can use. So what we need is our matrix, the counts matrix. So this one right here, and we need our samples file. This is just going to tell it to do it on a log2 scale with some specifications for the actual heat map, and this means compare replicates. So to do this, we just copy and paste all of this here, and it's just going to run it. It's going to read in the matrix file. It's going to do the QC for our 0 hours, and it's going to do our QC for the 6 hours. Okay, so now we have it. Right, it should be somewhere here. PDF, there we go. So 0 hours, rep compare PDF, rep compare PDF right here. Because we are a little bit tight on time, we're just going to look at all of these files a bit later when we run everything, so you can see how they look like. There's 2 more things that we're going to do. So compare replicates across samples. If we want to look at not just bioreps within a sample, but all of them together, we will run this. So we again have our matrix, some heat map parameters, our samples file. Again, it's going to be done on a log2 scale. We have our counts per million counts. And this is just to tell it, okay, give us the sample correlation matrix this time. So if we run that, again, it's going to go into R. It's going to read in the matrix. It's going to give us information. If this null device is here, it means that it worked. We do ls, and now we're going to have some more files here. So there is a PDF right here, and also the corresponding tables and data as well from that. And then the third thing we can do is create a PCA plot. Again, as I mentioned on the presentation, we are going to do 3 principal components, but you can change this number to anything you want. 2, it needs to be at least 2 or more. Copy, paste. Or again, there's something somewhere. Yeah, here, min row sums. Boom. Okay, ls, and now we should have some more files, including another PDF called PR-comp principal components right here. Okay, so to look at these files, we can now look at those PDF files here. So we need to transfer them to our computers. One way to do that is using CyberDuck for people that have Windows, or we can use STP. So for Mac users, I'm just gonna write something like PDFs, doesn't matter. Right here, so I created a file. I'm gonna remember where all these PDF files are. So PWD, they're right here. I'm gonna copy that just so I know where it is. I'm gonna open another tab. So this is my local computer, and I'm gonna use STP. So what I'm trying to do is secure copy from Hoffman to my local computer. First, I'm gonna make sure that I'm in the PDFs folder that I created. So that's CD, it's gonna be on my desktop, and PDFs. So it should be empty. So I secure copy. First, I put in my username and my path. So SCP Braber Hoffman, colon, our path to wherever our data is. And then we're going to use the wildcard, give me everything that has PDF at the end of the file. And we're just going to space, we're going to transfer it. This means in my current directory, which is going to be the PDFs. Enter. Then it asks me for my password. I'm not sure I put the right password. Let's see. No. And now it's just going to transfer all the files to my local computer. And then we're going to just have a brief look. So they should all be here. So we have for 0 hours, we have our summer of fragments. It tells us how similar they are by doing this MA plots, and then how the replicates are correlated to one another. And this happens per condition. So we have one for 0 hours, and we have the same one for 6 hours right here. Then what we did next is compared the conditions. And this is our correlation matrix. So these conditions based on this information are pretty similar, but still they separate quite nicely. So the 6 hours is in one bunch and 0 hours is in another bunch. If they're mixed up, that kind of tells you immediately, oh, maybe I mislabeled my samples, unless you expect them to be very, very similar to one another. So it's a good quality check. And then another thing that we did is look at principal component analysis. So you would want all of your same colored ones to be piled together and these to be separate. So as you can see here, because our data is quite small, they don't really match one another. So these would be the 0 hours, these would be the 6 hours. They're still a bit separate, but this data like this would not be amazing. And this tells you, okay, maybe this one is an outlier and maybe I should remove it from my data set from my downstream analysis because it might mess up my differential expression. So that's all kind of to do the quality checks there. Okay, so then the last step, we unfortunately won't have time for everything, but the last step that we're gonna do is perform the differential expression. Because our data is too small, I actually provided some data to be able to run differential expression analysis properly. So if you go ls, inside this data for DE is some more data that we're going to use. So if you go into this data for DE and press ls, you will have a samples file that I created. So it just has more replicates, more different conditions, and it has this count matrix and a TMM expression matrix as well that we're going to need. So what we're going to do is copy and paste this in. There's, again, a few things that we need to correct. So here we give it the path to our script. We need to input the matrix. We need to input the method. So it's going to be edgeR, and we're gonna give it the samples file, and we press enter. What it's going to do now is it's going to do pairwise comparison for each of these samples. So it's doing 0 hours versus 1 hour, and then it's going to go through it one by one. So 0 hours versus 6 hours, and all of the ones that we have. So this might take like a minute or so until it goes through everything. And then as this is running, we will just perform 2 more things for clustering to look at the PDFs again. So we will do the heat map to look at some genes that are differentially expressed and setting up a p-value, so we won't use anything that's below 0.0001. And we're going to do a cutoff for fold change to be at least 4 times different, for the genes to be at least 4 times different. And it's going to give us a table, but also a visualization of our data. And then after that, we're going to also do the clusters by using this program right here. So this script right here. Using data that's going to practically come out of this first command. And this is just another parameter to set up how you want your cluster trees to look like, how big you want the clusters practically to be. And this can be modified as well within the software. The most important thing I would say is just to try it out and see how it works. Which one works best, I guess. This is almost done. I think it probably ran 1, 2, 3, 4, 5. Yeah, this is gonna be the last comparison. Six hours versus our 1 hours. Yeah, this is the last one. We have, after we do this is going to be pretty fast. There is another part that has to do with identifying coding regions. We're not going to finish this by noon. I'm happy to stay longer if you want us to go through this as well. But if you want to try this on your own, you're more than welcome as well. Maybe we do that. The instructions are right here how to do that. So you go into the software, you get the transdecoder, and then you just run 2 very easy commands on the Trinity FASTA file. Okay, so this is done. ls. We have a directory created here called edgeR and some numbers. We need to go inside the directory. And we have a lot of different files. So we have the differential expression results for each of them. We have the volcano plots. We have the count matrix. We have the R script that ran this. So really a lot of information. And then now we're going to do the clustering. First, we're going to do the heat map. So we're just going to do copy, paste, and make sure we don't have any spaces here. So it's now doing the clustering. And the second thing we're going to do is to do the clustering with the tree. Again, we have a little bit of an error here. Space. There we go. If you press LS, there's going to be a lot of files now here. So we have PDFs and tables that correspond to those PDFs, and we have this right here. This matrix R data. It's just, we're going to go into it just so you can see what it is. Here we're going to have the plots in this PDF file. So let's very quickly look at this. I'm going to do it just on my computer. So we copy this path. We go back to my terminal here. And I'm just going to put the path right here to get the PDF for the plots. So my cluster plots, and then we're also going to download the PDFs from this directory right here. So let's just take that one. And again, I'm just going to remove the path for the RNA-seq workshop and add this one. Okay, give me all the PDFs that you have here. So we have the volcano plots and the heat map. And now we're just going to pull this back. They should all be here. So these are the cluster plots. And here we have 8 of them, and they just give us information about different paths. So it's quite useful if you want to look at, oh, I am actually interested in how this 6 hours, when only this 6 hours is affected. So you would maybe look at this cluster. And there is a corresponding table, of course, to all of these genes. These gray ones, those are all genes. And then we have the volcano plots for each combination of the pairwise comparison. Again, all of these correspond to some tables, really a lot of information that you can get out. And then this is, I think, the last one. And this is the heat map that we created. So we have some downregulated and upregulated genes. And this is all kind of a visualization of, what potentially can I explore? And then you move on from this kind of analysis to look at probably some protein level Go term enrichments and focusing on some specific genes of interest. Okay, let's just bring back this last slide to kind of summarize what we covered, although the last part was a little bit rushed. So practically, the summary of the Novo transcriptome assembly goes something like this. We have our FASTQ reads that we clean. We assemble them through Trinity or another assembler of our choice. In Trinity, we get the output of a Trinity FASTA file. And then we do 2 things. First, we assess the quality of the assembly by doing a lot of different options, such as detonate, Busco, transrate, looking at the protein coding genes, and assessing the read content and the length statistic. Then we can, after we determine that our assembly is good, we just do the post-assembly analysis, such as abundance quantification with some of these tools. We can do the QC of our samples, followed by differential expression analysis. We can also detect the coding regions that we can then input into Trinotate to get some more information of potential Go term enrichments and things like that. So this part of the path is actually pretty similar to a normal RNA-seq pipeline, whereas this part is quite specific to our Trinity de novo transcriptome assembly. And I think with this, I would like to close. We're already 4 minutes over. I apologize. If you have any questions, feel free to ask them. If you don't, feel free to email me. I will update all the information of on the slides and on the worksheet, so you have the final information, and it's written in a way that you should run it just on your own. If you run into any issues, feel free to, as I mentioned, to email me. And I sent the quiz in the chat, but I will also send you an email after this, after we finish. And I would just ask if any of you are students that are taking this for credit to please fill it out by the end of tomorrow. And that is it. Thank you so much for signing up for this workshop. I was very happy to teach you. I hope you learned something more about how to perform the Novo assemblies and why they are used. And I hope you got a little bit more familiar with using Hoffman as well. And that's it. Any questions? Any final things you would like to share? You are welcome. Thank you very much for attending.